Good morning. My name is Dr. Ben Newman, and I study coronaviruses. And once again, good morning. The next question is from Sasha. Yeah, how you doing? And for those who don't know, Sasha is what I would call an actual immunologist, not like me playing one on YouTube, but not very well. So, all right, let's see the question. Let's see. Hello. The vaccines by BioNTech, Pfizer, and my former university buddies at CureVac. Nice. All right. Yeah. Are all based on RNA technology, as you know. Yeah, this is like the modern version of what used to be called a DNA vaccine. This was a very spicy hot topic in the 1990s. And the RNA version is like a short term, but a little bit more direct way to do the DNA vaccination. So, yeah. And I think they end up putting a whole lot more in generally than they used to. Let's see. You sound as if limiting the antigen size, like which Pfizer have done, would still work for an antibody response, but may come at the cost of a T cell response. All right, let's read to the end. I guess I agree in principle, but if I remember my immunology correctly, and Sasha almost certainly does, yeah. There's no reason that effective T cell responses cannot be made against parts of the spike protein. Totally, yeah. Moreover, vaccine induced secondary T cell responses from antibody dependent processes eventually feeding into the T cell arm of the immune response, like epitope spreading, could also protect in the longer term. Yeah, I think so. My concerns come from two little points, and you can let me know what you think of those. One is in looking at the results that Pfizer BioNTech have put out so far. They have not looked at all at T cell responses except to say that the total number of T cells goes down, which suggests maybe some kind of selection, but that's all they did. They looked for, what regular alpha beta T cells, or maybe it was for CD3, something like that. Yeah, just all of them. And showed that there were fewer after vaccination. Yeah, it's just sort of an "Are we done yet?" Yeah, kind of approach. That could be a couple of things. I mean, if we're taking the worst possible view, this could be them sort of steering around a rock and saying, you know what, we don't have anything there. Let's just focus on the antibodies. The other thing it could be, though, because I'm assuming these are competent, smart people behind this. That seems like a reasonable assumption. I think I wonder if this is a deliberate sort of marketing strategy decision that the fastest way they can do the tests and get them out is to only focus on the antibodies and pretend that T cells may or may not exist, but we don't care either way. That is, I don't know. Yeah, those are the explanations that seem to make the most sense, but yeah, in the absence of any data, I don't know. The other side, and we'll say it concisely, and then I'll try and unpack it a little bit, is that given that presentation is HLA restricted, which for regular people, your immune system can only show certain things that they can grab onto. Or rather, your cells can only show certain things they can grab onto to the immune system. Yeah, there's so many things wrong with that sentence. But, and depending on which versions of these little sort of grabbers you're born with, you either will or won't be able to present certain things very well. And so what I'm thinking is that just in principle, given that everybody has this weird mix of the grabbers, which are called HLA or MHC class 1s, and 2s, yeah, there's going to be a very wide variety of sort of ability to show any part of this thing. And so if you've got more little pieces that you could be showing, then I think the immune system has a better chance of finding something it can grab really well. I don't know that that's necessarily the case, and you ought to be able to get a really hot, very directed response from T cells if you manage to put this into the right person who has the right set of those HLA grabbers. Yeah, but that is just logic. I'm not basing that on any paper. That's just, I don't know. That's my sort of worry at the back of my mind. And I think well, yeah. So that was the main two. The third worry is that there are already some mutations in the receptor binding domain, which is the part that Pfizer have gone for, and some of these are widely circulating enough that I think this is going to be the vaccine that has to be updated first. I don't know what the process is going to be for that. I don't know where they are in that process. But again, I'm assuming competent, very good at their job individuals, and I bet they're going to figure it out just fine. Yeah, as the virus changes, I think they may have to go to a blended strategy where they don't just put in all receptor binding domain from the one strain that they're using, where they maybe have a couple of different strains in there, like a modern flu vaccine. I don't know that we're to that point yet. They're all still pretty close, but we probably are to the point where somebody out there with some weird kind of HLA is going to, swing and miss at this thing. And we are seeing some escape from neutralizing antibodies in several recent studies. So that's what I'm thinking. I'm thinking they've built a thing that is high risk, high reward, and maybe a little bit brittle if the virus shifts, not least of all in response to this. But yeah, I don't see any reason why what you're saying is not right. I'm pretty sure it is right, and I'm pretty sure you know this stuff better than I do. So yeah, I look forward to it, and I hope that in the fully published phase 3 we get a little look at some of the T cell responses, because what they've done is really simplified the biological problem. Instead of having to look for through the entire virus for all those potential little parts that the immune system could recognize, they have cut it down to, what is it, about 200 amino acids? Yeah, total. Yeah. Which is a little bigger than a standard epitope, but, not that much bigger. Yeah. So there we go. That's kind of the outline of, the Pfizer vaccine. And that said, if you got some, I would totally take some right now. I think it's great that we've gone so quickly and got something that seems to actually work a little bit, because coronaviruses were considered a very tough nut to crack before in terms of vaccine strategies. People have tried a lot of things over a lot of years, and they have not worked out well in general. So there you go. All right. Thanks very much. This has been Ask Dr. Ben.